Imagine for a second that you're the architect of a medieval kingdom. You've built this incredible fortress.
Okay.
You've got high stone walls, a deep moat, and you have highly trained guards stationed at every single gate. It's impenetrable, that fortress. That is your body's immune system.
It's a perfect defensive machine, yeah.
Right. But now, imagine you take a pill for a headache, just a standard everyday medicine, and suddenly, for no apparent reason, all the guards fall asleep.
Oh, no.
Or maybe even worse, they get confused, turn around, and start burning down the castle from the inside.
And that right there is the absolute nightmare scenario for drug developers.
Yeah.
You're trying to cure a patient, and in the process, you accidentally leave them defenseless.
Yeah.
Or you turn their own body against them.
And avoiding that specific nightmare is exactly why we're here today. We are unpacking the safety net that stands between a new drug and, well, an immune catastrophe. We're doing a deep dive into the ICH S8 guideline on immunotoxicity studies for human pharmaceuticals.
And I want to flag something right at the top here. If you are a toxicology student or a grad student or, regulatory professional listening to this, pull out your notebook.
Seriously?
Yeah, consider this your audio study guide.
Yeah.
We know this material comes up on exams constantly because it relies on a very specific logic flow. So our mission today is to walk you through that logic so you can just ace those questions.
Exactly. We're going to break down the flow charts, the confusing acronyms, and the specific traps that usually trip people up. But before we get into the how, we have to define the what. When SA says immunotoxicity, what are we actually hunting for?
So, per the guideline, immunotoxicity is defined pretty broadly. It covers unintended immunosuppression, which is what you mentioned, the guards falling asleep, lowering resistance to infection or tumors.
Okay.
But it also covers immunoenhancement.
Which sounds like a good thing. Enhanced sounds positive.
In marketing, maybe. In immunology, no. Immunoenhancement means an exaggerated response. So this leads to hypersensitivity, allergies, or autoimmunity. But, and this is a massive point for anyone taking an exam, you have to know what is excluded from this document.
This is the exclusion list. If you see these on a multiple choice question about S8, they are the wrong answers.
You got it. This guideline does not cover general drug-induced hypersensitivity, so standard allergies. It also excludes drug-specific autoimmunity.
Huh.
But the biggest exclusion, the one that catches everyone, is the type of drug.
Right, because when I hear immune guidelines, I immediately think of those big, fancy biologic drugs we see advertised on TV for arthritis or psoriasis.
And that is the error.
Yeah.
S8 is for small molecules, your standard chemical pharmaceuticals, aspirin, statins, that kind of thing. If you are developing a biotechnology-derived product like a monoclonal antibody, you're in the wrong room. You need to be looking at ICH S6.
That is a critical mental partition to make right now. Small molecules go to S8.
Biotech goes to S6.
Don't mix them up. Okay, let's get into the mechanics of the document. When I first looked at this, I just assumed that because the immune system is so vital, regulators would demand a huge battery of specialized immune tests for every single new drug.
You would think so, wouldn't you?
Yeah, like you'd have to run every test in the book just to be safe.
But that would be incredibly inefficient, expensive, and it uses a lot of animals unnecessarily. S8 is actually a masterpiece of efficiency. It doesn't say test everything. It operates on a cause for concern model.
It's an investigation. You only call in the SWAT team if there's a credible threat.
That's a great way to put it. It's a filter system. You start with the basics, and only if you see smoke do you go looking for the fire. The foundation of this whole process, the first filter, is something called STS.
STS, Standard Toxicity Studies.
Right. These are your bread and butter, repeated dose studies in rodents or non-rodents. You're running these anyway to check for, like liver toxicity or kidney issues. SHS says, while you're doing that, look closely at the immune system.
Okay, so let's play investigator. I'm running a standard 28-day rat study. Not doing anything fancy yet. What am I looking for that screams immunotox? What are the clues?
The guideline gives you a checklist. The first place you look is the blood work, the hematology. You're scanning for a drop in white blood cells. Maybe total leukocytes are down, or, specific players like lymphocytes or neutrophils are missing.
Leukocytopenia or lymphopenia?
Yes, or even the reverse. A massive spike in counts could signal inflammation. But blood is just the circulating traffic. You also need to look at the bases where these cells live. You need to weigh and examine the organs.
The big two here being the spleen and the thymus.
Crucial organs. If the drug is causing the thymus to shrink enzymatic atrophy, that is a massive red flag. But S8 gets even more granular. You also have to check the lymph nodes and the bone marrow.
Okay.
And here's a nuanced point that separates the pros from the rookies. You have to check the lymphoid tissue that drains the site of administration.
Wait, let's unpack that. So where I put the drug into the body changes where I have to look.
It has to. If you're testing an oral drug, checking the spleen isn't enough. You need to look at the Peyer's patches in the gut or the mesenteric lymph nodes. Those are the first responders.
Makes sense. If the invaders are coming in through the front door, you check the guards at the front door.
Exactly. If it's an inhaled drug, you check the bulb bronchus-associated lymphoid tissue.
No.
If it's a nasal spray, the NALT. You look for damage where the drug concentration is highest.
Okay, so we have blood counts, organ weights, tissue structure. Is there anything else in that standard package?
2 more things. Serum globulins. If your globulin levels in the blood are shifting and the liver and kidneys are fine, it suggests your antibody production might be compromised. And finally, the most practical indicator of all, simply looking at the animals.
Are they getting sick?
Exactly. Are the treated rats getting pneumonia while the control group is fine? Are they developing tumors faster? If the animal can't fight off a standard cage infection, you have a functional immune problem, regardless of what the microscope says.
Okay, so that's phase 1, the STS filter. Let's say I find something. Maybe the white cell count is a little low, or the spleen looks a bit heavy. Do I immediately panic and order the expensive specialized tests?
Not yet. Now you move to the brain of the operation, the weight of evidence review.
Okay.
This is where you sit down with all the data and act like a judge.
This is step 2. The guideline lists 6 specific factors for this review. And honestly, for anyone studying this, memorizing a list of 6 dry regulatory terms is a nightmare. So we've hacked this a bit. We came up with a mnemonic.
We did. It's SPISDC.
SPISDC. It sounds like a secret government agency.
I'm with the SPISDC.
It helps it stick, right?
Yeah.
Let's break that down because understanding why these matter is way better than just memorizing the letters.
Let's do it.
The first S is what we just talked about.
Yeah.
STS findings.
Correct. How bad was the damage in the standard study? Was it a blip or was the thymus completely destroyed? You have to weigh the severity.
Then we have P pharmacological properties.
This is asking, did we do this on purpose?
Yeah.
If you are designing an anti-inflammatory drug or a leukemia treatment, you expect to suppress the immune system. That's the pharmacology.
Ah, okay.
So in that case, a drop in white blood cells isn't a safety signal. It's proof the drug works.
Right. Context is king. Next is I, intended patient population.
This is huge. Who is taking this drug? If it's for healthy people with a minor headache, your tolerance for immune risk is basically 0.
Right.
But if your patient population is, say, advanced HIV or cancer patients, people who are already immunocompromised, the risk calculation changes completely. You have to be much more careful because they have no buffer left.
Okay. That's number 2. Structural similarity. This feels like profiling.
It basically is chemical profiling. Does your new drug molecule look like another drug that we already know is toxic to the immune system?
Yeah.
If your drug is a chemical cousin of a known immunotoxin, regulators are going to be suspicious right away.
Guilty by association.
Pretty much, even if your early data looks clean.
Okay, then we have dedisposition.
That's just a fancy word for where does the drug go. When you dose the animal, does the drug distribute evenly or does it accumulate specifically in the spleen or bone marrow at high concentrations?
If it's hanging out in the immune headquarters, that's a risk.
Exactly. Why is it gathering there? And finally, C for clinical signs. If you are already in early human trials, are the actual patients showing signs of infection or immune issues? Human data always trumps animal data.
So that's the SPI, SDC. You take all these factors, you put them on the scales, and you weigh them.
And the guideline is strict here. A finding of sufficient magnitude in just one of these areas can trigger additional testing.
Just one.
Just one. Or, smaller concerns in 2 or more areas. It's a cumulative case file.
All right, the jury is back. The weight of evidence says guilty, or at least highly suspicious. We have a problem. We need to verify if this drug is actually breaking the immune system. What is the test?
If you are S8 compliant, you are almost certainly running a TDR.
TDAR, the T-cell dependent antibody response. Why is this the chosen one? Why not just count the cells again with better equipment?
This is the most critical concept to grasp for this entire topic.
Yeah.
Counting cells, which is called immunophenotyping, is just a census.
A census.
It tells you who is there, but having soldiers on the battlefield doesn't mean they can fight. They might be paralyzed or blind or, asleep.
So the census doesn't measure readiness.
Correct. The TDR is a functional assay. It's a war game. You take the animal and you inject it with a harmless foreign invader.
The guideline mentions sheep red blood cells.
Yes, SRBCs, or sometimes a protein called KLH keyhole limit hemocyanin, comes from a sea mollusk. To a rat, sheep blood or KLH is totally alien. Its immune system should see it, sound the alarm, and start manufacturing antibodies to destroy it.
And the TDR measures how many antibodies get made.
Exactly. And think about the biology here. To make an antibody against a new threat, a macrophage has to eat the invader, present it to a T cell, the T cell has to activate a B cell, and the B cell has to turn into a plasma cell. It's this complex relay race.
So if the TDR result is low, if no antibodies are made, it proves the chain is broken somewhere.
It proves the system is functionally broken. That is why it's the gold standard. It catches a failure anywhere in that pathway.
Now, you mentioned immunophenotyping using flow cytometry to count specific cell types like CD4 or CD8 cells. The guideline mentions it, but treats it differently.
It treats it as a secondary tool.
Yeah.
Flow cytometry is brilliant science. It gives you a detailed breakdown of the cell populations. But S8 classifies it as a non-functional assay.
Non-functional.
It's great for diagnosis, for finding out which cell type is missing, but for the primary risk assessment, regulators prefer the functional proof of the TDR.
It's better to know the army can't fight than to know exactly how many privates versus sergeants you have. Let's move to the real world. We have the theory, but how does this play out in a drug development meeting? Or a tricky exam question. I want to look at a couple of scenarios.
Let's do it.
Scenario 1. You are developing a chemotherapy agent. It's designed to kill rapidly dividing cancer cells. You run your STS, and surprise, surprise, the bone marrow is depleted, and white blood cell counts are down. Do you need to run a TDR?
What does your gut tell you?
My gut says no. I mean, we know it's killing the cells. That's the mechanism of the drug.
And your gut agrees with the guideline. This is what we call exaggerated pharmacodynamics. If the toxicity is a direct expected result of the drug's target, you generally do not need to run specific immunotox studies.
You don't need to kill a bunch of sheep blood cells to prove a cell-killing drug kills cells.
Exactly. It's redundant, and frankly, ethical committees wouldn't like using animals for a known outcome. The STS tells you enough to warn the doctors.
Okay, that's the easy one. Now give me the curveball, the stress trap.
Yes. This is where careers and exam grades are made or lost. This is the most common pitfall. Here's the physiology. You give a rat a very high dose of a drug. It makes the rat feel terrible. It loses weight. It's in pain. It's stressed out.
And biology kicks in. Stress releases glucocorticoids.
Cortisol.
Right? High levels of cortisol are naturally toxic to lymphocytes. They kill white blood cells and cause the thymus to shrink. This is a natural stress response. It happens in humans, too.
I see where this is going. A researcher looks at their data, sees a shrunken thymus, and says, oh, don't worry about that. The drug isn't immunotoxic. The rat was just stressed.
It's a secondary artifact. We can ignore it.
Right.
The stress excuse. It's the classic
Right.
get-out-of-jail-free card.
Yeah.
And for years, people used it liberally. But S8 slammed the door on that. The guideline states that you cannot simply assume stress. You have to prove it. You need what they call compelling evidence.
We need receipts. What constitutes a receipt for stress? Physical proof in the adrenal glands. The adrenals produce the cortisol. If the animal is truly stressed enough to melt its thymus, the adrenal glands should be enlarged, hypertrophy, or show increased activity under the microscope.
So if I have a shrunken thymus, but my adrenal glands look perfectly normal.
Then your stress argument is dead in the water. You have to treat that thymus finding as real immunotoxicity and investigate it.
That is a crucial distinction. Stress is not a default assumption. It's a diagnosis of exclusion.
Exactly. If you write, it's just stress on an exam without mentioning adrenal weights, you will get the question wrong. Guaranteed.
One last practical hurdle, timing. Let's say I have a drug that needs a TDR. When do I have to have that data ready? Can I wait until I'm filing for approval?
Too late. The guideline is specific. Additional immunotoxicity studies should be completed before Phase 3 clinical trials.
Why Phase 3 specifically?
Think about the scale. Phase I and II are smaller. They're controlled safety and efficacy checks. Phase 3 is the big 1,000s of patients. It's the exposure phase.
You need to know if you're going to crash their immune systems before you give the drug to 5,000 people.
Exactly. The only exception is if your target population is immunocompromised, like that HIV group we mentioned. In that case, you need the data even earlier, probably before Phase I, because the risk is immediate.
Okay, We have covered a massive amount of ground, from the flowcharts to the biology to the regulatory pitfalls. Let's distill this. If a listener is walking into an exam room in 10 minutes, what's the cheat sheet version of this deep dive?
Let's break it down into the 7 core pillars of S8.
Pillar 1, scope.
S8 is for small molecules only, no biotech, that's S6, and no allergies.
Pillar 2, the first filter.
Always start with the standard toxicity studies. Your STS, check hematology, organ weights, and histology.
Pillar 3, location matters.
Don't just check the spleen, check the draining lymph nodes, Peyer's patches for oral drugs, BALT for inhalation.
Pillar 4, the decision.
Weight of evidence. Remember SPI, SDC. A finding in one area can trigger the alarm.
Pillar 5, the test.
The TDIR is king because it is functional. It proves the system works. Immunophenotyping is secondary because it's just a census.
Pillar 6, the stress trap.
Never blame stress for thymic atrophy unless you have evidence of adrenal hypertrophy.
And pillar 7, the deadline.
Finish the studies before phase 3.
Boom. There it is. If you can explain those 7 points, you own this topic.
You do. And looking at it this way, you realize this isn't just paperwork. It's a sophisticated screening algorithm.
It really is. Before we sign off, I want to leave the listeners with a thought that struck me while reading this. We've spent this whole time talking about suppression, the drug weakening the immune system.
Which is the primary focus of the guideline, yes.
But the definition also included immunoenhancement, boosting the system too much. The guideline admits that testing for autoimmunity is really hard and not fully standardized yet.
That is the frontier. We are getting better at making drugs that tweak the immune system. I mean, think about cancer immunotherapies where we deliberately take the brakes off. As we get better at turning the immune system on, the risk of accidentally triggering dormant autoimmune diseases, lupus, rheumatoid arthritis, it goes up.
Yeah. A rat model doesn't easily show you that a drug is going to give a human lupus 3 years down the line.
Exactly. S8 deals well with the shields down problem.
Yeah.
But the shields too high problem, that is the wild west of toxicology right now. I suspect the next generation of these guidelines will have to grapple with how we predict autoimmunity before it happens in patients.
A terrifying but fascinating problem for the next generation of scientists to solve. That wraps up our deep dive on ICH S8. Good luck on those exams, verify your adrenal weights, and we'll catch you on the next one.
Stay curious.